Okay, so again, for those of you that did not have a chance to review that webinar, it's quite long. It's actually a 4-hour long webinar that we did for University of Thrace in Greece. And the presentation was also done by Dr. Gus Kasoulas, who is at the LSU School of Veterinary Medicine and has a lot of expertise in vaccine design, antiviral drug design, and very knowledgeable on those topics, and a lot of other molecular biology related to the vaccine design process. We also reviewed some of the bioinformatics approaches to a variety of topics that are going to be covered in this program. And we will, of course, do a lot more in detail, hands-on overview of some of those topics. So again, just to kind of remind everybody, what are we trying to get out of this program? We're trying to help all of you see how data available about this COVID-19 pandemic can help us understand what is really going on and analyze this data, which is a variety of genomic resources about the viral genome, as well as some of the analysis of host response. As you go through some of these topics that we highlight, we will refer to a number of different COVID-19 related publications, understand them, what are they really trying to tell us, how did they arrive at those results, and to try and understand how to replicate similar analysis once we find the relevant data. And the overall objective is also to understand key principles of vaccine and drug design. So in today's session, as a hands-on hands-on review and preparation of adapting some of these principles that we've covered up until now to a project of your own, we're actually going to be looking at NCBI virus, which is a database that we already started looking at, where we can search by virus and find a number of relevant coronavirus genomes. We can also see a number of different proteins and compare them to other viruses and other proteins as well. So we'll talk about that and do some hands-on and discuss how some of you might come up with a hypothesis that you can actually explore using some of these data sets. So as I mentioned, we do have the recording of the webinar that we had on Friday. So instead of our regular session, we actually did this webinar. The webinar was hosted by University of Thrace, and it was really interesting for me also to listen to Dr. Gus Kasoulas from Louisiana State University School of Veterinary Medicine. He did talk about some of the published work that he and his colleagues did on coronaviruses in bovine coronaviruses, as well as his experience working with coronaviruses today, as they are in the process of a number of different projects related to vaccine design based on the spike glycoprotein. I also had a chance to present a part related to bioinformatics, where we talked about important aspects of genome variation between different hosts, as you can see on the screen, and as they apply to evolution in the human population, starting from January 2019 or December 2019, when we have a lot of genomic data available on these genomes. But we also touched on some of the important topics related to vaccine design from a data perspective. We talked about antiviral drug design from a data perspective. So a lot of interesting material. And the link is in chat, so you can take a look. We also posted on the forum. So please try and review some aspects of that webinar. I think it's very relevant for this program. Okay, so now I'm going to pass it on to Beepsha, just to briefly speak about some of the progress for the two different programs that we have on our main online course and the LVRN group. Beepsha.
Yeah.
Would you like to
Thank you, Ilya. Am I audible to everybody?
Yes.
Okay. I'll start sharing my screen. Just give me a minute. Okay, I think everyone can see my screen right now. So I
Yeah.
just went through a few of the people who have been doing really well with the courses. So this is the progress tab. And if you go to the forum and under the topic of top performers, I just added a few people. We have seen that last week I'd added Ankita and a few others. So this week we have Benjamin and Adekunle and also Harnish. They have been really doing the courses and we see a lot of progress there, and we would request that everybody else also start doing the courses. So you can see here, since last week, Ankita has even more points. Now she's on 6210 points. And I know that you joined right now. So if you would please update your profile, that would be really great. So I just want to quickly show everyone how to update your profile if you are still confused about it. So what you do is that you go to your profile section. If you click on it, there is an edit button that comes up. So you can go to the edit button and there you will have different fields. So this is the button, right? And you see that there are a few other options. You can change the profile picture, you can change your cover image. So anybody else who does not yet have a profile photo, you can go ahead and update that and carry on with your courses. And also I see here, Shalini, you have been doing a lot of these courses, but again, your profile picture is not updated. So you can either add your own profile picture or you can go ahead and put in any avatar of your choice. And now to get to the certificates. Now this is something that we get a lot of questions about, like how I can get the course certificates that I'm completing. So you go to the courses again here, and under the courses, there is a section. So you see there are different sections right underneath. So there's quizzes, assignments, and certificates. If you click on certificates, you should be able to see all of the certificates right around there. And also, if you are having a confusion of why my name in the certificate is not appearing correctly, it is because your name is not updated, right? As we see here that, there is a period sign between the name, and this is also not his actual name, but just the username. So you have to update your full name so that it's reflected here. As Shalini's name is already updated, you can see that in her certificates, it is reflected. So that is how the entire system is linked together. So please update your names and update your profiles, because we keep on sharing them with our community, and it would be great to connect with each of you as well and learn from you about your research interests. And also moving to the LBRN forum, I also added here, I'm seeing that SAFE is doing really well, as well as Leela and Tyler has started. So you can look at their courses, the courses that they have completed. And you can actually see everybody's progress. If you go to the progress tab, you will all have that in the forum. You can actually look at the progress tab and see, how each of you are doing. If you would want to, like, say, for example, when you start a project, how you would want to collaborate with somebody. So we can all see that from here. And just another thing that I wanted to add is this, that we keep on sharing a lot of resources also outside of the forum page on our Facebook and LinkedIn channels. So we have over like 9,000 people here on Facebook page. It's kind of just loading for me. So if you would want to be connected here, you can connect with us here, I think. And this is our group in Facebook. So if you're interested, please connect with us so that we can add you to the group. There we share a lot of different resources, not just from Pine Biotech, but other things as well, like other relevant resources. And same here for our LinkedIn page. So you can see that this is our LinkedIn page. So anybody who is connected with me on LinkedIn, I keep you adding to also our LinkedIn group. And I'll be leaving all these links in the chats so that you can easily get on there, and you can add me, or you can add Mohit or Ilya in your contact list, and we'll be adding you to this page. So thank you so much, Ilya. Back to you.
Okay, great. Thank you, Deepsha, very much. There's a question there from Anurag. Can you walk through how we can complete a certificate? so I can address that. Again, when we have some of the, many of these programs, in fact, are going to be based on different types of materials that are available online, and not just what we show in the sessions themselves. Okay, so I'm just going to quickly show that. Okay. So in the program, right, we have the syllabus and also courses. So let's take a look at the syllabus here. You can see that some of these participants that Bipsha was highlighting, they have completed the courses, and you can see the courses under your profile, or you can see all of the courses right here. So if you are interested in understanding at a deeper level, or you might be confused by some of the terminology, you can go into this section and you can explore some of these courses. After completing these courses, you should be able to see that certificate in your profile. Okay, so hopefully that clarifies the question. Okay, and those of you that have reached out to us about questions or technical issues, we are getting back to all of you. We saw quite a number of different questions come up, and we are going to be addressing some of those questions. Bipsha has been responding by email, so do make sure that you have her email, which is marketing@PineBio, added to your contacts. We've seen a number of participants who have complained about not receiving the emails, but then later when they check, they're actually in the spam folder. Okay, so thank you, Bipsha, again for reviewing the progress. We addressed some of the questions. So briefly, let me review what we've covered in the past several sessions. Again, last session we had the online webinar. But before that, we talked about a number of different topics, including detection of viral genomes in animals and patients using next-generation sequencing, and mapping those genomics data sets onto databases of viral sequences. And so in the first session that we spoke, we spoke about metagenomic and viromic or viral sequencing. We talked about sequencing of bacteria and viruses that are present in host transcriptome. And then we also discussed how you can take a sample, map that sample onto the host genome, and then take unmapped reads and map it onto a database. There is an example in Metagenomics 1 that we asked you to take a look. And we did that to really understand how next-generation sequencing short reads, as you can see right here, can then be translated into actual full genomes. And these are the genomes that we're going to be looking at today on NCBI virus. We talked about a number of different pipelines, for example, visualization of mapped reads. And specifically, we talked about visualizing those reads taken from host genome. So here, what you can see on the screen is mapping of reads onto the COVID-19 reference genome. And you can see how reads are actually much more abundant here at the end, right? And one of the reasons for that is because this is taken from the host, from the human genome. And so the data preparation technique, which looks for poly-A mRNA sequencing, it's going to give you these kinds of skewed results, but you are still able to detect the presence of the genome, the COVID-19 genome, in the two samples that were infected with COVID-19. And in this sample that was not infected with COVID-19, it was mock infected, we do not see the genome at all. We can also annotate the genome, right? And here we see the different reads, or the different genes, sorry, of the reference genome. And that type of annotation helps us understand which genes have more coverage in terms of that COVID-19. And we discussed about finding these different types of data sets on SRA and understanding how they were sequenced and understanding what exactly can we find in human, for example, cell lines, right? And here's another example that we did for bat host. And you can see how in bat, you had two different types of samples. One was amplicon, one was whole transcriptome, total metagenomic sequencing. And you can see how the results are very, very different, right? So we saw here how we have, just a few reads, and here we have much more reads aligned to the reference genome if we use that bat rad G13 genome, right? Not the human one, but the bat rad G13. And we can also use this pipeline of mapping on the database. We took the NCBI virus database and mapped the reads onto the reference database to understand what types of viruses can we detect. So as we covered a lot of information in these sessions, and then we had the webinar, and the webinar was really kind of broad, covering a lot of many different topics, I wanted to pause here and make sure that we're not leaving many of you behind, that we're really making progress with the topics that we've outlined in the program, and we're not confusing you with the abundance of information about these different types of topics, and your questions are not going unanswered. So if you have a question, again, we've been trying to reply to a lot of the questions that have been posted on the forum. But if you do have any questions, I'm happy to answer those right now. And in fact, I'm going to go, as you're posting your questions, I'm going to go to the forum just to kind of see some of the discussions there and comment on those. Okay, for example, Shubham was saying that he is not able to visualize it correctly, but unfortunately I wasn't able to understand exactly what the comment was about. So please, whenever you post your question, make sure that you post the question with explanation of what exactly you are having trouble with. There have been questions on some other things, like, for example, variant viewing in the IGV. So I replied to that question. And the question was, after you do Genomics 1, there's an analysis of some genomic data, and you have to visualize mutations, and you can visualize those mutations in integrated genome viewer. So how exactly to visualize those variants? So please, I'm waiting for your response there. Sravia, in this specific topic, okay, let's see what was posted there. And again, if you have any other questions, feel free to add those on the forum. Okay, yeah, so here, again, the question was about accession numbers. So how annotation of accession numbers work. If you try to run a pipeline with annotation of genomes, sometimes it doesn't work. Sometimes it's because of the settings. And so make sure that you follow the instructions exactly. Adekunle. And if you have a question about a specific pipeline, then please post your pipeline URL. So let me just show you as an example. Let's say I have a question about this pipeline. You can see that this is the pipeline URL, and it has an ID right here. So for every pipeline that is submitted by you, I can actually take a look at this pipeline through the admin interface and see all of the settings that were done to make sure that I understand where the technical problem happened. So if you have any question about a pipeline, I will be able to reply and look into your settings. Okay, so a good question. Sravia Angara is asking, how can we extract the HTTPS of the SRR samples from the NCBI website? Okay, so we don't generally allow you to just start analyzing a bunch of data by uploading large data sets. You do have the ability to analyze public domain data sets, however, and we gave a few examples of projects where we provide you with specific data sets that you can practice with, right? So for example, we talked about uploaded data sets, which are FASTQ data, right? Now, the reason we're doing this is because there are limited resources that are shared between all of the participants of different programs. So if everybody starts running, hundreds of samples from NCBI, everybody is going to experience a dramatic slowdown, so that everybody is actually going to be very limited as to how far you can get with your analysis. So that's why we're asking everybody to, first of all, try the examples that we provide you with. Those examples are typically going to be on data sets that we have validated that they work, and they have predictable outputs from those analysis pipelines. Once you get to a certain level, and we monitor your level, we monitor your progress, as you have seen Deepsha explain, we monitor your progress in terms of point system, right? So after you get to a few thousand points and we see that you know what you're doing, you will be able to analyze your own data sets. We will explain in a later session how to get SRR samples from NCBI. And on purpose, we're pausing this because FASTQ files or SRR files are going to be much, much larger than the genomic files that we will look at today. Because today we're going to look at, again, how to do multiple sequence alignment and how to download those FASTA files, first of all, which are assembled genomes from NCBI virus. Okay, so Servia, let me show you again how to do this. I'm just going to take one of these just to explain. Okay, so if you get a link like this, right, you copy it, and then you go to areas of analysis. Okay, and then Mrudula is also saying, I need the SVL links from session 3 to run RNA-seq pipeline. I could not run the pipeline previously due to not being able to open the links provided. Okay, so I'll show you again. We do post those on the forum. So let's take a look. Here we have RNA-seq data. And these are SE, right? So you can see that these are single end. And we want to map them onto the reference genome that we upload. Not one of these, but one that we upload, right? So here you select our genome and you keep it SE. When you do file upload, you see that we are using SVL links, not HTTP links, but SVL links. So you just paste it right here. When I paste it right here, how do I know that this file actually got uploaded? Let me upload the organism here real quick. Okay, this file you actually upload directly from your computer. Okay, so I'm going to use this reference genome. It's a FASTA file. Okay, so the FASTA file is uploaded. I do group selection. If I see it right here, that means it was detected correctly. Okay, I can do that or do cancel no groups. And now I can just do bowtie 2G and visualization. And after I click on end, okay, you can click on run and run pipeline. Okay, so how to find these files? Again, we will go back over here on the forum. And on the forum, we should have session data files and presentations. Okay, the same for those of you on LBRN. Let's go back to the forum. Yep, I will address that question, Benjamin, also. Okay, so here you can see some of those links posted. So you download right here all of the data, right? And then you have some of the data sets inside. Okay, so let's first of all talk about session 3 files, right? So how do you actually download these files and what's inside these files for those of you that got a little bit confused by this? So if you click on here, okay, you can see you get this zip file. You unzip this file and you have a number of different files inside. You have bat-rad-g CSV, right? bat-rad-g GFF, bat-rad-g genome bat-FASTA, right? Then you have this GFF, this FASTA file, and also you have this TXT. So this TXT that I shared, okay, which screen am I sharing here? Okay, yes, I am sharing this screen, great. So this is where I took one of these samples, right? You just copy and paste it, or you can actually upload it as bulk upload. Okay, so let me show you that again, and I'll explain the difference between bowtie 2G and bowtie 2T. So again, these are SE files, right? So they're single end, and I'm using my own genome, not the reference genome, but my own. Okay, somebody is talking a lot on Skype. Let me close that. Okay, so here I have the reference genome, and I'm going to upload FASTQ files, and they're in single end format. Okay, Benjamin, Rodula, Sravia, again, I'm doing this for you. If you have questions along the way as I'm explaining this, please stop me and I will be happy to address those questions. File upload. In this zip file, I have actually 3 different examples of file uploads that you can do. One example is using direct SVL links. So this is called a direct SVL link. If I have many of these, I can either add more and paste multiple, just like I have from here. I can copy and paste them in here. Okay, or if I have many and I don't want to upload all of them, I can do add files. Sorry, URL bulk upload. So URL bulk upload will be, okay, this list of links. When I do choose, okay, I have there 3 files, remember. So I'm going to, after I upload the organism, which was the this one, SARS-2, you can see that it detected that there are 3 different files. So these 3 different files contain reads from 3 different samples, right? I don't need groups here. And what am I trying to do in terms of mapping those reads onto the reference genome? I click on Start, and I do have these 2 options, bowtie 2G and bowtie 2T. Now, how do you know what the difference is? The difference, you can see right here, it says mapping on genome, and here you can see it says mapping on transcripts. T stands for transcripts, G stands for genome. So what are we doing here? Benjamin, please tell me, are we mapping on genome or on transcripts? What do you think? Genome, right. So which one would you choose? Bowtie 2G or bowtie 2T? G, excellent, okay. So we choose bow tie 2G because we're mapping on the genome, and then we can visualize the mapping, okay? So that was example one, right? Example one, we use files that are already preloaded on the platform. Now, instead of preloading the files, right? So let's take a look what else is inside here. We also have this CSV file, right? So the CSV file, and this is more related to, let's see, whose question was it? Sravia, okay? Sravia, please let me know if this is making sense for you. So we have here a BAT-RAT G13 CSV, and you can see right here are the links to the data sets on NCBI. So you can see download paths. So in this example, how do you actually get these on here? Again, I'm going to do our genome, file upload. Instead of going to URL bulk upload, I'm going to go to NCBI run table upload, and I will add the files from here. Okay, so I'm going to take that CSV, choose. Again, I have to upload the organism, which is the reference genome I want to use. So I can use again this reference genome. You see, those SRA files have been uploaded. Now, what are these SRA files? These SRA files are from a bat. They're metagenomic sequences of bats, okay? So that is the second option. The first option is when we have preloaded files on the server. Second option is when we have them directly from NCBI. There's a third option too, which is directly from your computer. And you saw how we can upload a file from your computer using the direct upload files button. Okay, so Sravia, does this make sense? Did that answer your question? Okay. So, okay, excellent. And a question for Marudala. Marudala, did that answer your question about the links? All of the links, again, where are they located? On the forum, I pasted here session 3 files, and they are right there. You download them, and inside you have all of these different files, and they are explained. All right, excellent. So hopefully this explains to everyone how to navigate the different resources and files and projects to be able to replicate the analysis from your own computer on your own account. Again, please do this in the context of the sessions that we do and in the context of the assignments. Okay, and again, Wesley is asking, how do you assemble or make the CSV files for NCBI upload? So I think I showed it in session 3. I don't recommend, I don't want everyone to go out and, get hundreds of samples right now and start uploading them to the platform to run them. So please don't do this, but I will show you how to do this since everyone is asking. So if I go to NCBI, again, what's going to happen if many of you start doing this, at the same time, it will just slow us all down. So let's wait off until we get to the stage of projects, and we'll actually tell you specifically if you're, ready to do that. So please hold off on this, but I'll just show you as an example. So let's say here, I want to analyze some data sets that are on the SRA database for NCBI. And here I will just look at, what's available. So you see DNA metagenomics of adult fruit bat kidney. Okay, have no idea what they did here, but let's just take a look. So here they have extracted with QIAGEN RNA-easy kit without use of DNA samples or ribosomally reduced using, okay, so somehow they prepared this on a lane of Illumina HiSeq. This is just one sample, right? So I want multiple samples. For example, I can go to all runs, okay? And here you can see, actually they only have one. Let's go back. Let's go to this. Okay, 4 SRA experiments. Okay, so here they have some 4 SRA files. Here they are, 4 of these files. I go to send to and click on file. And here, instead of format, I do run info and create file. So this is that CSV file that I gave you. Okay, it contains the run ID and then the download path. Okay, so Wesley, does that answer your question? All right, excellent. All right, any more questions about what we've done up until now? Okay, so if there are no more questions, let's take a look. As you know, there are many available data sets about the novel coronavirus genome. And there are 2 major questions that we're going to explore in this program about the way that this virus originated in animal hosts, eventually through a single or perhaps multiple spillover events, it went into the humans. And within the human population, that virus continued to evolve. So what I showed on Friday in the session that we did, in the webinar that we did, is that you have this excellent website that allows you to understand how the novel coronavirus spreads throughout the different zones of infection. You can see here how the virus spread historically, and you can also see how the genome mutates over time. So in fact, if you play this right here, you can see how the genome, again, may be related to the number of samples that were available, may be related to how the virus started to mutate as it spread into the different regions, right? It doesn't really tell us why. It just shows us this is the data that we had at that moment of time, right? So as you look through this animation and you can see how the virus spread throughout the world, you are going to start wondering, how are the different genomes in different locations different from each other, right? So up until now, we looked at how are they different in the host as opposed to the human, different animal hosts opposed to the human. Here we are seeing how the virus mutates over time in the human population. So what we see here is how the virus separates into different clades in this phylogenetic tree. As it separates into the different clades in this phylogenetic tree, we can see that that is because of accumulation of mutations in different proteins. These are the different viral proteins on the COVID-19 genome. Now, all of this data is actually available. So a lot of this data that you see right here, you can actually download all of the data. You can go here and do download data, and it will tell you what kind of data is available. And a lot of the publicly available data is actually going to be on NCBI virus, a resource specifically dedicated to the hosting and sharing of these genomic resources. So how can we find a number of different genomes? Again, these are going to be genomes in typically FASTA format, they're fully assembled genomes, and compare them between the different locations. Again, a quick question here from Adekunle. Can we get the URL for this page to view in real time the mutations? Seem the next strain? Yes, so this is the link. Okay, so here you just click on play, you can play it, you can see the individual genomes right here. So again, a lot of these are on the NCBI repository, and you can search here by virus. So you click right here, and I'm just going to also put this link in the chat. And the purpose of this session is to show you how to actually download the sequences yourselves, to do it on your own, and to practice doing it, to build a phylogenetic tree on and produce a table of mutations, right? So reproduce some of the aspects of what you saw on Nextray. So please follow along with me, right? So I'm just going to share a few steps. They're simple steps, but they require for you to make decisions along the way on what exactly you are going to be looking for, okay? So please follow along and try to tell me if you don't understand a specific step or you don't know where we are, so we can stop and address any of that confusion. Okay, so did everyone get to this page where I am on NCBI? Okay. All right, excellent. So a lot of you are there. Okay, so here, what do we search for? We search for the coronavirus, okay? So if you just type in corona, you will see here the different options start to appear. And I'm just going to focus on the severe acute respiratory syndrome coronavirus 2, or the SARS-CoV-2, because it caused COVID-19. Okay, so I'm just going to click there, and you can see that right here under the filter, we have severe acute respiratory syndrome coronavirus 2. Okay, so here you can see that there's 8,133 nucleotide sequences available, but not all of these sequences are the same. As you scroll through this table, you'll be able to scroll sideways, see that they are different lengths, different geolocation, and different hosts. So let's refine the search a little bit. Instead of just looking for any sequence, we are looking for only complete sequences. So you can see that the complete sequences are 5,569. Okay, so let me know if you are able to see the same thing. Okay, so here you can see that I got 5,569. And now let's start asking specific questions from this data. So one of the questions that I can ask is, what is the difference between viral genomes in, let's say, Washington, state of Washington, and let's say state of Louisiana, let's say New Orleans, right? So here I can go to geographic region, and I can say USA. So there's 4,000, right? So a lot of these genomes are from the US. So if I click on, yeah, I believe this is just by country. Okay, so let's go to USA. And as you look at USA, some of them just say USA, some of them will say specifically a location in USA. So this, unfortunately, this interface is not, 100% easy to use. As you saw, I couldn't search here for Washington, for example, and I can't find here New Orleans. It will take me some time to actually find those. So instead of trying to figure out how this works, what I'm going to do is I'm going to go to download. And here, before I start downloading individual FASTA files, I'm just going to download this table in a CSV format. So I'm just going to click on Next, download all records. And here also offers a number of different column titles for what I can include in my CSV dataset. So you can look at accession, which is this ID right here, release date. Is it important for me to have a release date? Not really. So I'm just going to eliminate this. I'm only looking at humans, so species or host here is also not very important to me. Isolation source, maybe, for example, lung or gut or something like that. Collection date, definitely important. Geolocation, definitely important. I will also include GenBank title. That will contain some important information there as well. Okay, and I did select for only complete sequences, so I'm going to illuminate this as well. Let's download. So when I download this CSV, for anyone that was not able to download this CSV, please let me know. But I'm assuming that if you haven't stopped me, that everybody is following along just fine. Again, we're going to open the CSV in Excel. Okay, so I'm just going to show you what it looks like once I open it in Excel. Again, you can use any kind of a table visualization and manipulation software. Just for me, easier to do it in Excel. So let's take a look at what is available here. Okay, we have different levels of data completeness for each individual sample. But the most complete one is actually going to be right here under, this is GenBank title. Why? As a requirement of compiling the GenBank sequence, you are required to add any information that you have. And you can see here that even though some of this information is not present in geolocation, just stated as USA, some of this information is going to be present in here, and we'll be able to see it for the majority of the samples. Okay, so let's take a look. Now we can search for New Orleans. Okay, and here we have a number of different locations in Louisiana. Okay, I am in New Orleans, but we can see a number of other ones. So you can see Metairie, Kenner, Hammond, Slidell, etc. So all the familiar places. Let's look for New York. Okay, so here's some in New York. Okay, so now you can search for different locations. How do you take samples? Okay, so up until now, is that clear? Does anyone have any questions about what I'm doing? Okay, excellent. So how do I go from actually, okay, Anuja, no questions, okay. So how do I go from here to actually getting those FASTA files, the genomic sequences? Well, the genomic sequences are actually all located under this accession number, right? So for example, if I take this accession number and I go to NCBI, okay, just the main NCBI, and I search for this accession number, okay, you can see that this is the nucleotide sequence that comes up right here. And I have the FASTA file, or this is the GenBank accession number, okay? So what if I want to have multiple? There's a site on NCBI that is called Batch Entrées. Okay, so which is a way to get files for all of the provided accession numbers. Okay, so because it is easy to work with this table, as we saw, let's just go back to New Orleans. Okay, so here I have in New Orleans 1, 2, and 3, right? Or I can say, okay, well, here I have all of these samples from Louisiana, right? So I have, I can take them, for example. So I'm just going to take these from Louisiana. I'm going to copy them over here. And, you know, where do we want to compare it to? Let's compare it to Michigan. Okay, it doesn't really matter. So let's just take some equal number of samples from Michigan. Okay, so here we have maybe a little bit less. Okay, so here we have prepared some table. I'm just going to copy this over as well. So I have here all of these accession numbers. If I want to download them all in a single file or two different files, I'm just going to create one for all of the ones from Louisiana, which are these, and paste them in here. Make sure that you make it into a plain text. So now you have these accession numbers. Okay, so I'm going to save this in my data, and I will call this Louisiana FASTA. Then I'm going to create another one, and that's going to be the Michigan samples. Again, I'm going to copy these. I'm going to make them into plain text and save them as Michigan. Okay, any questions about this? Okay, no questions. Again, if you want to do it from India, how do you do it? The same process. If you wanted to, you know, specifically in Wuhan as opposed to Beijing in China, you know how to do that, right? So you just create these simple TXT files with just the accession numbers. Then you go to NCBI batch and trez. You can choose a file. So let's select where did I place that? Okay, place that in a different place. So that is right here. In section 3, data. Okay, so right here. So I'm going to choose my Louisiana FASTA file. Okay, and then click on retrieve. Can the files only be TXT files? Yes, these are going to be. In fact, you have here on some direction on how to use this. Okay, so it should be just plain TXT. All it contains are the accession numbers. Click on retrieve. You can see that I found 15 records. So I'm just going to click on retrieve records for 15 of these. Okay, and here you see that they're listed out. I want to download all of them. So I'm going to click on send to, complete record, file, and then format and FASTA. Create file. Okay, so remember this is 15 genomic sequences. So I can call this, these were from Louisiana. And let's open and take a look at what's inside. Okay, inside, if I search for this symbol, I will find 15 of these different sequences. Okay? Now let's do the exact same thing, but we will do it with the Michigan ones. So instead of LA FASTA, I'm going to choose the Michigan FASTA, also just a TXT, just contains the accession numbers. Have 16 of those. Okay, and here they are. Send to complete record file format FASTA, create file. Okay, so as I am doing this, you're probably thinking, okay, how is this useful for me? So can you suggest, some of you, suggest what types of comparisons would you think would be interesting to make? Okay, think about it for a couple of minutes and answer in the chat box, what kinds of comparisons would it be interesting to make based on the different types of metadata that we have about these genomic sequences? What do you think? What kinds of comparisons would be interesting to make? Okay, so Manish is saying mutation. Okay, find mutation of spike protein, mortality versus genomic difference. We can answer, where did the virus arrive in particular location? Great. Two different types. Okay, so great ideas here. Genomic difference in varying geographic locations. Yes. Okay, so as we can see, right away you have some ideas, right? So in different geographic locations, okay, probably many of you know, for example, some locations had 0 deaths from the virus. Some locations had many deaths from the virus. Does that mean that the viral genome was actually different in those locations? Does it mean that the population was very different in that location, right? So there are many different questions that we can start asking. If we have an idea that the population was very different, right? So let's say, I believe it was in Thailand, 0 deaths. Obviously, Thailand is in Asia, right? And one of the questions that was suggested here, where did it come from to Thailand? Did it come from China? Did it come from Australia? Did it come from Japan, right? If we have a question about the human population, that factor, can we answer that question with these FASTA files? Yes or no? Can we answer the question about human population factors using these genomic data sets? So Benjamin is saying probably. What does everyone else think? Can we answer a question about the human factor using these FASTA files? Okay, Kasturi is saying no. Okay, and you can add an explanation why. Why do you think so? So what are these genomic sequences? If you have the metadata, not about the human factors if the data is not available. Okay, so what is this data that we're looking at? These are genomic sequences. Okay, so these are genomic sequences of the virus, right? So these are coronavirus sequences. We don't have any data here related to the human factor, right? So we don't know what is, who was this isolated from, right? None of that data is available in this file. So when we have a question, we can find some data that can answer that question, or we can find some data that cannot answer that question, or we can find some data that can definitely eliminate any other types of concerns we might have. For example, if we think that this was a combination of human and genomic variation of the virus, we can make sure that the virus itself is not different in any major way. Okay, so it's good to think about what your question is before you just jump into analysis, so that your analysis is actually relevant to the type of data that you have. So let's take a look. Now that we have these FASTA files, right? We have two different locations. We selected them arbitrarily. What type of an analysis can we do to actually ask the question, how are these two locations different between themselves? So we can look at a number of different questions. We can analyze the data in terms of their sequence, right? So we can take a look at, let's upload these data sets that we just downloaded, the FASTA files. Okay, please follow along so that you also have the practice. Okay, we upload those FASTA files. We can put them into two different groups, Michigan and Louisiana. Okay, so here we want to use a GenBank file for framing codons. As you remember, the individual mutations might have more impact if they actually change the amino acid. So let's add a file here for the genome. So let's see, do we have the genome? We have it in one of the sessions that we used. So FASTA. Okay. Let me get it from another one. Again, how do I, let me actually show you how do I download the genomic sequence to use for codon. Okay, so that was the question. Let's go to NCBI. And we'll use SARS-2. And here we have the reference genome sequence for this novel coronavirus. And we're looking for coding sequences of codons, right? Because here you can see the sequence is in amino acids, and we so far have just this sequence, which is the FASTA sequence. So we can download this file, send to complete record file, but instead of FASTA, we will use GenBank. Okay, so the GenBank file actually contains the conversion between nucleotide sequence and amino acid sequence. Let's take a look at what we downloaded. It says sequence 3. Okay, so let's call this SARS-2 reference. Okay, so here we have SARS-2 reference. So let's upload this as our GenBank file for framing codons. We're going to use the SARS-2 reference. Okay, does anyone have any questions about this? Kind of, yes, it's kind of like an annotated genome. Yep. Okay, so no other questions. And now it says evolutionary time outgroup. Okay, what does that mean? When we do our analysis, evolutionary analysis between different sequences of genomes, we get an idea of how different they are in terms of their mutation. And we can get a sense of how might they be related to each other and link it to evolutionary time. In other words, if I have 100 mutations difference between them two, and I think that they needed maybe 10 iterations between the origin and the observed sequence that I have today, I can now give it a rate of mutation, but I still don't know how far away that was in real time. So, but if I have some references that I do have a very specific time of collection of those samples, and I can use that time to link with my evolutionary time, I can understand the distance between observed sequences and origin sequences, linking evolutionary time of mutation rate and real time in terms of years, months, etc. I don't have that right now, so I'm just going to skip. And it's also not relevant because actually I have the date of collection for each one in any case. So now I can start and do my multiple sequence alignment, converting it to amino acid sequence, and also using the select codon position. Okay, and the reason I want to do this is because I want to understand how the different mutations at the nucleotide level correlate with differences at the amino acid level. Okay, so let's do all phylogeny methods. I can keep it at nucleotide. I will still get both results because I have all of these selected. And I can also analyze the different trees that will be a result of these phylogenetic tree methods. Let's call it FASTA, Michigan versus Louisiana, COVID-19. Okay, I'm gonna run this pipeline. And by next time that we meet, we'll be able to look at some of the results that we got based on this analysis. But instead of doing exactly what I did, I want to ask you to start thinking about a question that you might have and you can ask about some of these available genomic data sets on NCBI virus. So you kind of saw the process, right? You can either find that directly in NCBI and you can have some general filters that you can apply to study different locations by their region or by their country, by genome completeness, sequence completeness, by maybe host and even date of collection or date when they were published. And you can start asking some questions, right? So what would be really great for the next time for all of you to have some sense of is how do your choices about the data that you want to select reflect, how are they reflected in the results of your analysis pipeline, and what can we ask about the meaning of the results that you get, okay? Instead of focusing on something that I got, focusing on something that you got, okay? So please try to do that, and I will post some directions on the forum. So again, for those of you that have not been able to view the forum, please review the forum to see there what is it that we are trying to do for the next session. Okay, so in our next session, this is what we did today. In our next session, we're actually going to look at the results of this phylogenetic analysis and learn about how this phylogeny can be divided up into specific groups of sequences. Again, you're welcome to look at some of the presentation from the last webinar. We talked about this. And here you can see some of those results that we're going to be looking at next time. These are different clades. You can see that originally there were only 3. Actually, originally there were 2, and then there were 3, and now there's over 1, 2, 3, 4, 5. But the G is actually separated into 3 other ones. So we actually have 1, 2, 3, 4, 5, 6, 7. 7 clades. So you can see 1, 2, 3, 4, 5, 6, 7. So there are some clades based on data, right? The latest data from 2020 06 16. Okay, so next time we'll take a look at the results in general, but also we'll take a look at some of your pipelines so that we can understand how those pipelines can be interpreted and what do they tell us in terms of the differences in the genomes of SARS-CoV-2. So that's for next time. For today, any other questions I can answer about the analysis that we did today and about finding those sequences on NCBI virus? Okay, so if there are no more questions, I'll be looking forward to seeing you again next time. Please let me know if you have any questions by posting them on the forum, and I'm going to be looking forward to answering those questions and looking at the results from your analysis. Thank you very much. We'll see you again soon. Okay, can we get human metagenomic data of affected patients? Yes, we will go through how to find metagenomic data of affected patients. I showed today how to search for NCBI on genomic sequences, but you can also look on NCBI for human metagenomic sequences as well. Okay, we will have a session dedicated to that. That's a byproject, exactly. Yes, Benjamin, that's exactly what you would do, is you would look for a bioproject on the project tab. Oh, yes, so we will work on an independent project. Right now we're organizing the data, right? So what's important is to find a data set that answers your question. Now you're in the process of generating your question. Once you know your question, we'll have a submission form. That submission form is going to be where you submit your project idea and your question and the data sets that you found. And once you have a good sense of what you're planning to do, you'll have the ability to work on your independent project. All right. Great, thank you, and looking forward to talking to you again soon.